I'm going to talk about plant nutrition, and I'm actually really, really pleased with like what Emily's talk was all about because I feel like you're all now much better. You have much better grounding in the sort of complex exchanges that go on with plant roots. And because plant roots and fungi and bacteria and what we call the rhizosphere, which is just sort of the area immediately around plant roots and includes all that stuff, it's really understudied. You don't learn about it very much at all. And that kind of pattern and neglect carries over to things like crop breeding and crop management. Like we think about crops as things that have leaves. We don't think about their roots when we're breeding for certain traits, for high yields, for sturdy growth. Well, we certainly didn't much in the past. We're thinking about it more these days, and we're researching fungi and we're researching bacteria. But up until recently, we haven't really thought much about, the roots of plants. So, all of that to say, you lot are going to be some of the most informed people on, below soil biology in Sheffield. Thanks for hearing 2 talks on it. So I'm here to explain how plants get nutrition and how bacteria tie into that and how we can sort of work on engineering this to our advantage to reduce reliance on fertilizers and things. So obviously, plants get carbon from photosynthesis, and there's other nutrients they need that aren't carbon that they have to get from the soil because generally speaking, they can't sort of digest their own food like we can. There are, of course, exceptions to this rule because it's biology. But, usually they need some kind of below ground exchange to absorb mineral nutrients. A lot of them form alliances with fungi. They have that symbiosis and they get their nitrogen, their phosphorus, and their potassium from the fungi, which are better adapted to both absorb it and to digest it. Because, these sort of the simple forms of minerals that our bodies use aren't just floating around freely in the soil. They're usually bound up in bigger, more complicated molecules, organic molecules, and rock minerals, and we have to get them out somehow. Fungi are really good at digesting, organic matter and rocks. They'll excrete these enzymes and break it down like we digest food, and they can use that to help plants. But there are also other ways that plants can acquire nutrients. I'm going to talk to you today about P specifically, because PhDs are specific. That's right, phosphorus, one of the most important elements of plant nutrition. I'm afraid I'm not talking about urine. That's nitrogen. I don't study nitrogen. However, phosphorus is really, really important. So you can sort of see, I've got a pointer. The form that the plants need is this simple little molecule. It's called orthophosphate or inorganic phosphate. And that's, the sort of phosphorus as they use it in their bodies. And phosphorus, as you find it in your body, is bound up in various larger molecules. So this is a phospholipid, like phospholipid bilayer, the cell membrane, which every single cell of your body has. Loads of phosphorus there. Glucose 6-phosphate, that's a part of the sugar cycle. Breaking down sugars, they put a little phosphorus on the sugar. And of course, your DNA, it's got phosphorus in it, right here. And, we all have DNA, plants have DNA, mushrooms have DNA, all living things have DNA, and it's therefore the soil's full of it in various stages of digestion. So why is phosphorus important, and why can't we just get phosphorus in this form and add it? That's what chemical fertilizers are, so we should really just be using those for maximum efficiency. After all, it's been shown, I haven't got the graphs to hand, but yields go like this when you start adding sort of phosphorus fertilizer, nitrogen fertilizer, rather than just organic manure. But as you can see, that's tiny. And the thing about really small molecules like that is they're easily washed away by rain. So you might have encountered, news articles or something on what happens when you have lots of fertilizer that is then washed away by rain. You get stuff like this. You get massive, the algae is also a plant. It's also starved for phosphorus fertilizer, and it's also doing really well from the massive addition of plant-available phosphorus. And that causes all kinds of problems. Waterways get clogged, fish die, it runs out of oxygen. It's just, this is clearly not the best solution to plants needing more nutrition. So instead, we're going to turn to biology. I'm specifically going to talk to you about bacteria, because what bacteria are really good at is digestion. They're so, like, if you think about our gut bacteria, we have loads of them. And if you don't have the right gut bacteria, then you will have problems digesting your food. You might even have like developmental problems with the way your guts grow if you don't have the right ones at the right age. So These Frankfurts. Sorry, I'm slightly under-rehearsed. And plants are very similar. They require the right sort of bacteria to break down these organic nutrients. And these bacteria do that by the means of enzymes. And an enzyme, for those who don't know, is like a little protein with a very specific shape, and it does a very specific task. I'm studying enzymes that break the bonds between, these little phosphate molecules and whatever they're connected to. There's a whole load of different enzymes that bacteria produce that can do this, but I'm working on a couple of specific ones. The Pho group, which are produced by Pseudomonas bacteria and many others, and Pathé, which is produced by some Bacteroidetes species, for those of you who care about bacterial classification. Who doesn't? So yeah, you can see the action of the enzyme here. It breaks off the little organic phosphorus and it releases glucose, or NPP. And that's, I'm telling you about NPP specifically for reasons that I'll show up later. But first, I need to talk about genes as well. So genes are instructions for making things. They can make, the proteins that make up your body, the structural proteins, the hair, the skin, the nails, the organs, whole new cells, and enzymes. We have loads of genes that are for specific enzymes. And some of them are only switched on when we need the enzyme to do a specific job. Maybe we're running out of protein digestion enzyme 15B, so your body, recognizes that it needs to use the gene that codes that enzyme to make more of it. And the bit that tells the gene to be used is the sort of switch. Most genes have switches right at the beginning. Some of them are, they're basically held down. They run all the time. The gene is produced over and over. Like your hair is always growing. The genes involved in producing hair are presumably always on. I'm not an animal biologist, so someone might need to fact check on that. But others, like the digestive enzymes or like bacterial enzymes, are only switched on if they're needed. It makes sense. If I was a single cell bacterium, I wouldn't want to waste energy making enzymes I don't need to digest food I don't want day in, day out. So bacterial phosphatase, phosphatase enzymes that release the phosphorus, are only switched on if there's not enough phosphorus for the bacteria. And that's not very helpful for us as plant enjoyers, because that means if there's not enough phosphorus for bacteria, the plant has probably already died for the lack of it. So we need bacteria that are going to lend a hand to the plant and are going to keep on producing these phosphatase enzymes while the bacteria doesn't need the phosphorus, but plant does. Is everyone with me so far? Okay, I'm seeing some nods. That's better than I expected at this point in the night. So we're going to move on to talk about the microbiome bit. I just wanted to show you this picture. These are all the same species. They're growing sort of different liquids, but they lined up nicely with their little plastic casings, and I got a good group picture. These are my children, everyone. I know. So, yeah, beautiful range of colors on these, but the important thing is that they're all found on the roots of wheat. That's where I got them from. I got a wheat plant, I grew it up, I scraped some stuff off, I put it on some agar plates that will kill all the bacteria except for the ones in the group I'm looking for, the Pseudomonas. And the reason I'm using those is because that sort of group of bacteria is quite well known for being quite good for plants among those who care about these things. And these have the perennial problem of not turning on their phosphatase enzymes if they're not really hungry for phosphorus. And I figured out, I haven't figured out, I'm working on a way around that with my supervisors, posited and has been used in biotechnology before, because I've explained what the problem is. The problem is they're not producing the enzymes. I got ahead of myself. The other problem, actually, is that even if you find bacteria that are great at producing enzymes, you can't just put them on a field and hope they'll take. There's other bacteria already living there. Usually, microbial inoculants work really, really well in the greenhouse. These bacteria are so good for plants. You grow plants with the bacteria, they're so much healthier than their peers. But when you apply these healthy bacteria to a real-world field, they just die. So what can we do about that? We can use plasmids. So this blue squiggle, that's the bacteria's genes. That's all the genes that belong to the bacteria. That's what they use to make their enzymes and to grow and to move and to develop and all of that. And then there is a plasmid. The plasmid has its own genes. It's made of DNA. It's the exact same DNA, but it's selfish. It's not part of the bacteria. It's more like a virus. It'll infect the bacteria. It will make the bacteria create copies of this plasmid, and then that bacteria will get real close to another bacterium, create something called the pilus, and inject some plasmids into the other bacteria. Sorry. And these plasmids are really important to humans because they're one of the main ways that antibiotic resistance gets around. You get some plasmids, it somehow grows a gene, evolves a gene that gives resistance to, methicillin, for example, and then it copies itself, and then it spreads into other bacteria. And then suddenly you've got a whole load of bacteria across different species that all have this methicillin resistance gene because of this plasmid. And then when those bacteria find other bacteria, they can infect them with that resistance. So the genes are transferred horizontally between bacteria in different groups, in different species, in different places. And that's a huge problem because that's how we get superbugs. But it's also really cool because, the plasmid's pretty small. It's a lot easier to edit a gene here than it is to edit one here without causing problems with the neighboring genes and with the regulation and with them being produced. If we look at sort of the biotechnology field where we're using E. coli to produce useful proteins and stuff, plasmids play heavily into that. I'm speeding up now because I think I'm running out of time. I'm about on time. So what I'm looking at is taking a gene for a phosphatase enzyme, and this phosphatase enzyme is unusual because its own bacteria, the ones found in nature, they produce it all the time. They don't switch it off if they've had enough phosphorus. They keep on using it. And we think that's because, to oversimplify a little bit, see here, you go from G6P to glucose. They're not interested in this bit. They're interested in this bit, and they're using that phosphatase enzyme to get this bit. So this can result in lots and lots of these phosphates being free, released, floating in the environment, being absorbed by the plants. And it's like, well, what if we just put it on the plasmid, put that gene on the plasmid, put the plasmid in all these different bacteria, and, turn them loose? Theoretically, this will result in vastly increased phosphorus concentrations around the roots of plants, and, much less fertilizer requirements, which is good. We make phosphorus fertilizer from phosphate rock. We're not going to get any more phosphate rock anytime soon. It takes billions of years to form. We found a new deposit recently in, I want to say Norway, but most of that's going to be used for electric vehicle batteries. So, we're going to need alternatives that aren't phosphorus fertilizer. And just to sort of give you a little demonstration, I wasn't allowed to take my bacteria out of the lab, but this is a time lapse. That yellow stuff, hang on, back again. Okay, so this is, when it's a liquid, it is clear. When this is a liquid, it is bright yellow. And if the enzyme is present and cuts off the phosphorus, it goes from clear to bright yellow. Okay? So this is a time lapse of about 30-40 seconds. But you can see two liquids that are the same color. I add the PMPP to both of them, but one of them, over the 30-40 seconds, goes a really luminous yellow. That's how fast the enzymes are working. That is how concentrated the enzymes are. It's the exact same species of bacteria. It's grown in the exact same conditions, the exact same amount of phosphorus and everything. The only difference is one of them has a plasmid. And that plasmid has the phosphatase gene, PAF-A phosphatase gene, and it is switched on all the time. Switch is held down. And, this is the kind of thing that makes people like me and my supervisor really excited. But I can understand that it is just a drop of liquid turning yellow. So you're going to have to take my word for it, and that's pretty exciting. So the other problems with my work, apart from, the fact that it's presumably not going to work as well sort of in real plants and real soil as it does in test tubes and vials. One, I am technically making genetically modified products. I'm making plasmids that have been edited by gene modification techniques. And the point of these plasmids is to spread as efficiently as possible through a world population in the soil, which, is pretty good at not having hard barriers in it. I think we can all sort of see the potential problem with this work leaving the lab, right? The government certainly is not going to greenlight this. Not sure about, what the ethics reviews would say. On the one hand, they don't, it's not the sort of ethical complications you get when you're engineering a human being, but there are still ethical questions that will be raised. I could argue that it's not a genetic modified organism because plasmids aren't classified as organisms, but I'm not sure how that would go down. The other problem is the same problem as when we introduce bacteria from, into a new field. It's turtles all the way down, right? Competition is a problem within the plasmid world as well as in the bacterial world. We can introduce bacterial inoculants and find they've been outcompeted by the locals within a week, and we can introduce plasmid inoculants and presumably find out they've been outcompeted by the local population of plasmids within the wild bacteria in the similar time frame. However, there's probably going to be ways around that by making the plasmids confer some kind of competitive advantage. Again, this would that level of engineering of ecosystems would raise a lot of ethical questions. So it's all, it's all going to be done in tightly controlled glass vials that get incinerated. But as a model system, I think it's very interesting. I think this has a lot of potential. And I think that is all I have to say, unless anyone has good questions. Thank you very much for listening.